Hello, my dear student. Today we are going to discuss autocoids. In autocoids, basically the histamine. Today we will going to discuss the basic about the histamine. I am Dr. Rishikesh from Department of Pharmacology, RG Sabka College of Pharmacy, Engineering, Nasik. The outcome of today's lecture, and at the end of the lecture, students able to understand the definition of autocoids, what do you mean by histamine, its biosynthesis, storage, release and metabolism, drugs or chemical that liberate and inhibit the histamine. So these are the outcome of this today's lecture. So autocoids, what do you mean by autocoids? Autocoids is basically a local hormone. It is a Greek word. It is a autos. If we break the word autos, self, akos, that is a remedy. So self remedy. It is produced by tissues or endothelial cell. After release locally, it act at the site or near the site of their release. So it is also called as a local hormone. So histamine, serotonin or bradykinin. These are the examples of autocoid. And their agonist and antagonist have clinical uses. So basically histamine is what? So histamine, hist, tissue, and which is available in the amine. In the tissues, what the specific amine is available, that is called as a histamine. It is a naturally occurring imidazole derivative. So if you see the chemistry part, here you find this is a heterocyclic imidazole nucleus with amino group side chain. So where it distributed, basically histamine in the skin, gastrointestinal tract, mucosa, lungs, brain, cerebrospinal fluid and bone marrow. So these are the various distribution site where the histamine is available. So histamine is a component of some venoms also and stink secretion. Means most of the after bite of mosquito, okay, or after a bite of any ant, so there is some secretion of histamine, and that histamine may cause urticaria like reaction or allergic type of reaction may be seen. Okay. So the next part, that is how the histamine get biosynthesized in our body. So that I already told you, histamine, here you find this structure, okay, imidazole ring with this side chain, ethylamine. Histamine is basically formed by decarboxylation of L-histidine. Okay, this is one of the type of amino acid. This reaction is catalyzed by the enzyme histidine decarboxylase. Histidine to histamine in the presence of this enzyme. Okay, so this is a biosynthesis. First of all, this requirement, histidine decarboxylase enzyme has a very wide distribution. Okay, this enzyme has a very wide distribution. Example, these are located in the most of the tissues, mast cell, GIT, mucosa, and certainly or some actively growing tumor in the body. Okay, this histidine is available, and some activation of these histidine decarboxylase enzymes are basically by some chemicals or some human infection, stress, nicotine, activators. These are the activators for the histidine decarboxylase enzyme. And if you want to find out its inhibitor, So alpha fluoromethyl histidine, which is used as a research tool, so that which inhibit this histidine decarboxylase enzyme. Sorry. So this is one of the important part where the histidine plays a very important role for the synthesis of histamine. Sorry. Mast cells. When we go next slide, histamine storage. Okay, where it get stored. Mast cells are the predominant site of storage for histamine in the most of the tissue. So I already told you hist, tissue, okay, and amine, tissue amine. Basically, in these mast cells, histamine is held in mast cell as a intracellular granular complex with an acidic protein and heparin of high molecular weight. Okay, so this it is an intracellular granule with an acidic protein, that complex, and heparin of high molecular weight, so that become macroheparin. It is also located in the blood. That histamine stored in the basophils. So basophil is one of the type of WBC cells, differential WBC cells. Okay, then histaminocyte, these are also non-mast cell in the stomach. Then histaminergic neuron in the brain, where the histamine is available or stored. The next part, that is the histamine release. How the histamine get released? Okay, from the mast cell, how it get released? Okay, that is a very important. So it get released by immunologic release, or chemical, or mechanical. So immunological release means there is some antigen antibody reaction. Obviously it should be there. Initial exposure to allergen leads to the production of IgE antibodies, which then respectively bind to their respective site, that is IgE specific receptor, which is present in the tissue of mast cell, present on tissue mast cell or blood basophil. Re-exposure to allergen, again, that leads to the cross-linking of mast cell membrane bound IgE. Okay, and that is once antigen attaches to the two IgE antibodies on the surface of the mast cell. So this union causes the degranulation of cytoplasmic granules and release of histamine with allergic mediators. So that is an immunological release. And this type of release, it also requires some energy and some ions like calcium. Okay, so histamine released by this mechanism is a immediate type of reaction. Okay, so this type of reaction is called as an immediate type of reaction, or type 1 hypersensitivity reaction. Okay, so it is a very simple antigen antibody reaction when re-exposure to again that particular antigen. So degranulation of the mast cell, and the degranulation of mast cell which releases the histamine, and that shows the allergic type of reaction. This is very simple. Next one, chemical and mechanical release. Chemical or mechanical mast cell injury cause degranulation of mast cell and histamine get released. So there are certain drugs like DTC, D-tubocurarine and radio contrast media directly replace the histamine from heparin protein complex within the mast cell by non-receptor action. After their exocytosis means what? That it's a process by some ions or some action potential arises in that particular vesicles, and that get released, or storage granules, and due to some action potential, or that get burst out, and that releases the particular chemical. So after exocytosis, the histamine heparin protein complex is exposed to extracellular cations, that is a sodium or cation, where histamine is displaced and released after cationic exchange with sodium or calcium. So this is a process of chemical and mechanical release, where this type of cations are involved, that is the sodium and calcium. Drugs like morphine, succinylcholine, DTC, radio contrast media. Okay, so that directly replaces the histamine from heparin protein complex. So that is the mechanism. So histamine is one of the several potent physiologic mediators that are released from the mast cell. Other substances such as serotonin, leukotrienes, they are also contribute to the overall hypersensitivity reaction and anaphylactic shock. Now next we will see the histamine metabolism. So histamine is metabolized by histaminase to imidazole acetic acid and by N-methylating enzyme imidazole N-methyltransferase, followed by monoamine oxidase B, okay, to N-methylimidazole acetic acid. Okay. So these are the very important histaminase to imidazole acetic acid and by N-methylating enzyme imidazole N-methyltransferase, followed by monoamine oxidase B. IMAA, okay, imidazole acetic acid is then excreted in urine as amino acid conjugate. Okay, so this is a simple histamine metabolism. Here I will just show you this diagram or this picture or this flow chart will give the idea. N-methyl imidazole acetic acid, N-methyl histamine to N-methyl imidazole acetic acid, imidazole acetic acid to imidazole acetic acid riboside. So these are two enzymes that involve. Okay. Drugs or chemical that liberate. Next one, the drugs or chemical that liberate the histamine. So previously I had told you morphine, DTC. So morphine, opioid analgesic, this muscle relaxant, trimethophan, there is a muscle relaxant, skeletal muscle, molipixine, it is antibiotic, succinylcholine. Okay, these are the anticholinesterase enzyme, hydralazine, dextran, polyvinyl pyrrolidine, detergent, substance P, bradykinin, radio contrast media. So all these agents, drugs, may cause the release of histamine. Then heat, cold, exposure to sunlight and X-rays, that also involve for liberation of the histamine. Proteolytic enzyme trypsin and chymotrypsin, then as well as venom also involved for the liberation of the histamine. Now next one is the drugs or chemical that inhibit. So beta 2 antagonist. So mostly the bronchodilator. So example adrenaline, ephedrine, isoproterenol. These are the agents that acts as a antagonist and which inhibit the mast cell histamine release. So mast cell membrane stabilizer, example that is the disodium chromoglycate and ketotifen. So these are the also mast cell membrane stabilizers. So these agents generally used for the treatment of asthma because histamine plays very important role for allergic reaction. The next one, negative feedback control on mast cell and basophil by the action of if release the histamine through H2 receptor. So H2 receptor basically present in the GIT or that is in the stomach, and that may also cause the acid secretion through the H2 receptor. So basophil and mast cell means basophil or mast cell that plays very important role during any injury or any infection. Their level get rise. Okay, and by this way we can diagnose that person suffer from such sort of infection. And lastly, thank you very much for your patience listening. And here in this 10 slides we have discussed what is autocoid, what is a histamine, how it get biosynthesized, where it's stored, how it get metabolized, how it get excreted, and how it get liberated and inhibited by different types of drugs or chemicals. That's all. Thank you very much once again. I hope you go through this video and just take the advantage of this. If you have any difficulty, you please mail me or just send the feedback. Thank you very much.